Well, hello and welcome to the first session of ICAAC Live for the 2012 meeting of ICAAC. That's the Interscience Conference on Antimicrobial Agents and Chemotherapy. We'll stick with ICAAC from now on. Coming to you from San Francisco, I'm Jeffrey Fox, your host for many of these sessions. There will be about 10 or 12 sessions altogether. Let me give a couple of ground rules for people on the internet. If you would like to ask questions, please use hashtag ICAAC or the ASM newsroom to send your questions over the internet. For those of you in the room with us, please raise your hand. Someone will come to you with a microphone, then identify yourself as to your name and institution and ask your question. I encourage you to, once we get going, to interrupt the flow whenever you like with your questions. You don't need to wait for an opportunity later on. Okay, so with those ground rules, have I introduced myself? I'm Jeffrey Fox, the features editor of Microbe, the monthly magazine of the American Society for Microbiology. And let me introduce my first guest, who is Danuta Skowronski of the British Columbia Center for Disease Control in Vancouver, BC, Canada. And she's going to be talking to us about some experiments looking at the flu vaccines and how they behave in ferrets. I think what we should do to begin with is to let you give us a several-minute summary of your findings, and then we'll begin to put them in context with a conversation, questions and answers from me and from others, either in the internet or in the room. So go ahead. Thank you.
Okay, thank you. Well, the experiment that we're reporting here at ICAAC is a follow-up to a series of studies that we did in 2009. And in 2009, several observational studies in Canada showed an unexpected increased risk of medically attended pandemic H1N1 illness in those who had previously received the 2008-09 seasonal trivalent vaccine. That was an unexpected and I have to say most unwelcome finding. And in the beginning, those findings, and it was 5 or 6 studies that in Canada that consistently showed that effect.
And a lot of people?
Well, it was about a 2-fold increase in risk for those who had previously received the seasonal vaccine of developing medically attended pandemic H1N1 illness. And in the beginning, those findings were considered and were labeled, frankly, a Canadian problem because studies in other countries showed mixed results. Some showed that there was no effect between prior seasonal vaccine receipt and pandemic illness. Some showed even protection associated with that vaccine. So it was quite a conundrum. There was quite a bit of skepticism about the findings. I should say that in follow-up, other studies have also shown a similar effect in other countries, including, I think, very importantly, a randomized controlled trial, the very best kind of study design in children in Hong Kong, as well as a Japanese study. And other animal studies have also shown an association between seasonal vaccine and enhanced disease, for instance, in swine, and as well a ferret study done by another group. However, there was still, I think, reasonably some uncertainty, some skepticism about the findings. However, since 2009, it's not possible to further study that association between seasonal vaccine and pandemic risk because seasonal vaccines since then have included the pandemic strain. However, we also felt that there was a scientific and frankly a moral duty to further understand, to do further investigations, to clarify those findings to the extent possible. That's important from a public health perspective in terms of maintaining confidence in our immunization programs. So we designed an animal study. You can't do human challenge studies for ethical reasons, of course, but it happens that the ferret model is an excellent model for studying influenza. It's the best model, in fact. And so we designed a randomized, double-blind, the ferrets didn't know what they were getting, but neither did the technicians nor the investigators, a randomized...
No, the ferrets weren't blinded, of course.
The ferrets didn't know what they were receiving.
They were not like the 3 blind mice.
Well, no, in fact, there was more than 3. There were 32 of them, and certainly none of them, and neither the investigators nor the technicians who were giving the vaccine or the placebo and who were following the ferrets, they did not know what the animals had received. And so we randomized 32 ferrets, 16 per group, to receive either the 2008-2009 seasonal trivalent vaccine or else placebo injection. And 2 things are important. In previous animal studies, we were quite surprised to see how few animals are typically included in animal studies. And if you want to show statistical significance, you certainly need more than a couple ferrets or animals per group. And also this was a randomized double-blind controlled trial, so that we were trying to ensure to the extent possible objectivity in evaluating the findings.
So, and let me just backtrack for a second with you. When you say the trivalent vaccine, this was the same vaccine that had been used to immunize the human population whose, those epidemiologic findings you were trying to mimic or sort through, I guess, not mimic, but so you were using that same vaccine in the ferrets, in some of the ferrets, to test what it would do when they were exposed to H1N1 flu, right?
Correct.
Okay.
Correct. It was the 2008-09 commercially available trivalent influenza vaccine, the product that was domestically produced in Canada, and we gave the ferrets according to the same schedule and dosage as was given to people in the immunization campaign.
Scaled to their body size.
No, in fact, we gave the same dose, and that's typical with ferret studies, yeah. We gave them the same dose as, in fact, usually with vaccines, you don't tailor the dose. That's something many people don't realize. You don't tailor the dose to size or weight. And so for the ferrets, we gave them the same dose and schedule as was given to people.
And you found?
Well, what we felt, first of all, the reason that we were doing the experiment was first to clarify, were our findings real? Because with observational studies, it's always true, those study designs are susceptible to bias and confounding. That's not to say that investigators are deliberately biased in their measurement of the findings or the results, but it's just inherent in the design because you are observing what's happening, and human behaviors can alter the findings in ways you cannot control. So the first was we wanted to see if in a randomized controlled study, the very best kind of study design, using the best animal model for influenza, if we could replicate the findings. And indeed, we did. The findings that we show are consistent with the increased risk that we saw in the human studies. And the second purpose of our study was to evaluate, well, okay, if it's real, what is the likely mechanism? Because I think many people doubted our findings on the basis that it was hard to come up with a biological mechanism to explain it. And people thought there's no biological plausibility to the findings. So our second objective was to clarify what is the likely immunologic mechanism. And currently, I think in the scientific community, very many now do believe that our results were real, and the debate is, what was the mechanism?
Okay, just for a second, let's summarize those results a bit with the ferrets. The ones that received the vaccine were became sicker, is that right, when they were exposed then to H1N1 flu compared to the ferrets that did not receive, that were not vaccinated. Can you describe that in a little bit more detail? How, in what way were they sicker? Did more of them die? Was this a fatal disease for them, or did they just, you had other measures of how seriously ill they were from the flu virus?
Okay, so in the human studies, we saw that there was an increased risk of medically attended illness, but not subsequently an increased risk of severe complications like hospitalization or death. And similarly, in the ferret model, we saw an increase in acute illness, but by day 14, after challenge with the pandemic strain, the ferrets had fully recovered. None died. However, compared to the placebo group, the animals experienced more severe acute illness. And we followed them on multiple indicators, clinical indicators, activity score, temperature, weight, for instance. We looked at nasal and lung virus titers, and we looked at lung histopathology. And what we found was that the animals in the vaccine group had statistically significantly greater weight loss by day 5 post-challenge compared to the placebo group. They also had a more rapid increase in nasal virus titers, but also subsequently more rapid decrease. That was not statistically significant. We importantly found that they had statistically significantly higher lung virus titers, and their lung histopathology score was worse than the animals in the placebo group. In other words, they had more acute bronchopneumonia compared to the animals in the placebo group. So on several indicators, all consistently in the same direction, with some significantly different, the animals fared worse in the acute period up to about 5 days post-challenge with the pandemic strain, but thereafter they recovered.
Okay, so we have something of a paradox. A vaccine that wasn't really designed for H1N1, but is designed for influenza, for other types of influenza, in this case made the animals more susceptible to a new strain of the influenza virus. And that's a bit odd. So you say you're looking into mechanisms to explain that. Maybe we should talk a bit about those mechanisms, because there does seem to be a paradox here of sorts, and one that is worth talking about, for sure.
Yeah, and I think you touch upon a very important point, and that is that our findings are unique to the pandemic context. They can't be extrapolated to other seasonal influenza experience. And I say unique to the pandemic context because in that context, what we saw was a strain that was very different, a major antigenic change from what was previously circulating and from what was in the seasonal vaccine. By definition, a pandemic represents a major shift in the virus, and this was a major difference in the pandemic strain versus what was in the seasonal vaccine. But it's very important because what it signals to us is that there remain important immunologic interactions between seasonal viruses and pandemic viruses when they emerge. And it's very important for us to better understand what that interaction is, because pandemics are infrequent occurrences, but there will be another pandemic. There will be more novel emerging viruses that we will possibly have to address this issue with. So it really is important for us to better understand that. I do think it's important to clarify that our findings are unique to the pandemic, and they do not mean that people should not get their seasonal vaccine. That's very important for me to emphasize, because pandemics are infrequent occurrences, but seasonal influenza recurs on an annual basis. It's a substantial cause of morbidity and mortality, and the seasonal vaccine substantially protects against that severe outcome due to seasonal influenza. But there, nevertheless, is something very important in that interaction with pandemic viruses.
And you did mention to me earlier, and we want to reiterate that the seasonal vaccine now the one for this year, 2012-2013, the upcoming flu season, does incorporate an H1N1 component so that the phenomenon you're studying in the ferrets has been addressed with that vaccine. The vaccine that's available now for humans to use is going to be protective in a way that the earlier vaccine was not protective against H1N1.
Correct, because the seasonal vaccine includes, as you say, that H1N1 pandemic strain now, so it is well matched. And what we're reporting are findings in association with dramatically mismatched, and I emphasize again, dramatically mismatched, as in the pandemic context.
So you were going to speculate some about the immunologic mechanism that might underlie this phenomenon you've been studying. Can we talk about that?
Yeah, sure.
Because I'm really interested in knowing how the earlier vaccine might have primed these animals for becoming more susceptible to a new type of flu strain. That's an important finding and deserves some intellectual exploration here. So
well, it really,
what do you think is going on?
It is fascinating, and it was fascinating to us as well. I think that this ferret study helps to clarify, point us in a certain direction, but as always with influenza, it opens up a whole bunch more questions as well. So right now, there are two dominant hypotheses in the scientific community to try to explain our findings other than bias and confounding. And of course, what's very important about this study as well is that it says that bias and confounding, which are inherent in observational designs, cannot explain away what we observed in 2009. So that's important. So now in terms of an immunologic mechanism, there's two hypotheses. The first is either that it was a direct vaccine effect due to the vaccine inducing low levels of cross-reactive antibody from the seasonal vaccine to pandemic virus, cross-reactive but non-neutralizing. And typically that's seen in an original antigenic sin type phenomenon, an antibody-dependent enhancement effect. So when you have low level of antibody that recognizes but doesn't neutralize, it can actually facilitate rather than prevent uptake of the virus into cells. And that's most typically seen in association with dengue. It has been
right,
the dengue effect.
Phenomenon, where if you have one form of dengue, then the next time around you get even sicker because you've seen some dengue and that somehow makes your immune system go crazy when it sees the next type.
Exactly, when there's a different type.
So there's a kind of a good framework for understanding. What's the other hypothesis? Okay,
so that's the possible direct vaccine effect through an antibody-dependent enhancement type of mechanism. But the second hypothesis is that it was an indirect vaccine effect. And the hypothesis is that the seasonal vaccine, which we know is very effective in protecting against seasonal influenza viruses, may have prevented people from becoming infected in the previous year or years with a seasonal virus. And it's recognized that infection due to seasonal virus provides some better cross-protection against new strains. So by protecting against seasonal virus so very well, the vaccine then blocked the cross-protection that could have come from infection. And that hypothesis has been referred to as the infection block hypothesis, or the temporary immunity hypothesis. You blocked the otherwise cross-protection from seasonal infection. But I can tell you that
Isn't that a little bit akin to the hygiene hypothesis in a way, that if you're not exposed as much to all kinds of different antigens, diseases, and things, your immune system doesn't get as good a workout. It's not in as good a shape as it might be if it had been more fully exposed to flu in this case?
I would say the hygiene, yes, you're right. And the hygiene hypothesis is a kind of extreme of that
Yes.
hypothesis. In this case, it's specific to influenza, and some very reputable groups, for instance, the Osterhaus group in the Netherlands, has in fact shown that seasonal infection will protect against novel influenza viruses like H5N1, and that vaccine that protects against seasonal infection will in fact block that cross protection. But I can tell you that
Well, let me stop you here, because I see that we have a question from the internet, and,
Okay.
and if I don't let those questions come in a expedited fashion, I get in trouble. So go ahead, Jim, if you would share that question with us.
Yes, this question actually comes from Helen Branswell at Canadian Press.
We're having trouble hearing you, Jim.
I'm sorry, this question comes from Helen Branswell at Canadian Press via Twitter. And she wants to know, the 2009 virus had an H1 hemagglutinin antigen. Would you hypothesize whether the effect would have been worse if the HA had been entirely novel?
if it had been entirely novel?
Yes.
Hi, Helen. It's a great question.
This is coming from a friend of yours? This is an insider question. Okay.
Hel-
Go
Hel-
ahead. It sounds like a tough one anyway.
Helen's she's a very reputable influenza reporter. She's well known amongst influenza scientists, so great respect. It's a great question because for antibody-dependent enhancement to occur, there has to be a precise balance, a certain balance of low level. If it's too high, you won't see it. If it's extremely low, you won't see it, the effect. So low level, cross-reactive, non-neutralizing antibody in a precise balance that facilitates uptake. So and I say it's a great question because the pandemic in 2009 was a unique pandemic in that it wasn't an entirely novel subtype. It wasn't an entirely new hemagglutinin protein on the surface. So there was still some recognition. It was an H1 virus, and H1 viruses have circulated since 1977. So it was an H1 virus that had a major antigenic change, but it wasn't completely novel. It wasn't completely new. So with our seasonal vaccine, we were inducing H1 antibody that could vaguely recognize it. It could recognize the HA sufficiently well to link to it, but the hypothesis being that it was at low level, non-neutralizing levels, sufficient enough to cause the kind of ADE, antibody-dependent enhancement effect. However, that's still a hypothesis, right? That we don't know that, and this ferret study certainly does not conclude that. What it does is it clarifies and points in the direction it suggests it was more likely to have been a direct vaccine effect, that the indirect mechanism is not necessary for these results, for that increased risk to have been manifest.
Okay. We have another question in the room, and if you would identify yourself and...
Dan Keller with Medscape. If I remember right, people who had more competent immune systems had worse outcomes with H1N1. So it seems like younger people, and essentially you've boosted an immune system to give them a better immune system, non-specifically, but still more reactivity. You had your bronchopulmonary effects, I guess, were more severe. So I guess the question is, number one, with any new vaccine, since you test it for neutralizing antibody and you can't do a challenge, how do you know whether it's going to be better or worse for the population? And you've been talking the whole time about neutralizing antibodies. Did you look at cellular mediated immunity, and what was the effect in your ferrets that way? And a third mechanism, which you didn't approach, was any sort of inflammatory response. Is that also in the mix?
Mm-hmm. Again, all really great questions. Again, I want to emphasize that our findings, both the human study findings and the ferret study findings, are unique to the pandemic context. So when there is a major change in the hemagglutinin from what we've previously seen. For seasonal influenza vaccine, where there are minor mutations, what we call drift in the influenza virus from year to year, those are small mutations. And the seasonal vaccine has been well evaluated. The safety and the effectiveness of seasonal vaccine is well established. And again, I want to emphasize our findings do not suggest you should not get the seasonal vaccine because of these results. So it's been well established. In terms of other markers of immunologic mechanisms, we did look also at cytokines, lung cytokine response, to try to get at that cellular marker, cellular mechanisms, and cell-mediated immunity. And what we found was that there was a trend toward higher lung cytokine values in the vaccinated ferrets as well at 5 days post-challenge for each of the cytokine markers that we looked at. Interferon gamma was so low it was not detectable, but for each of the other cytokines that we looked at, in particular IL-4 and IL-17, the trend was to be higher in the vaccinated group than the placebo group at day 5, suggesting that there may have been some inflammatory component involved. What was very interesting, however, was that by day 14 post-challenge, there was a significant decrease in the lung cytokines in the vaccinated group, but not in the placebo group. And that suggests that there may have been some kind of concomitant downregulation in the immune response. And perhaps that was mediated again through the high IL-17, IL-10. But again, those results, we're just beginning to scratch the surface of understanding this, and it's really our sincere hope that others will pick up the gauntlet and investigate the immunologic mechanisms in more detail. We've now been able to clarify the results appear to have been real, and that it appears to have been a direct vaccine effect, but now others need to look more closely at what the precise mechanisms may be.
Okay, good. We have 2 pending questions. We'll take the one from the internet first, and then we have a question in the room too, Jim.
Yeah, this first question is a follow-up from Helen Branswell. She would like to know, might the problem not have occurred then with an entirely novel HA? This is going back to her earlier question.
And just to remind people, HA is hemagglutinin. That's one of the 2 major antigens on the surface of the influenza virus.
Well, I want to point out that when we observed these findings in the first place, they were entirely unexpected. So it's only been a few years now that we found those observations, and we've been trying to follow up on them. So I wouldn't have thought that this was possible or would have happened at all a few years ago. So to then speculate would it not have occurred with dramatic is difficult. But I get your point, Helen, and I would say that my own opinion, my own feeling would be that if you have a completely different hemagglutinin like H5 or H7, rather than an antigenic change within the same subtype, you may not see that. But who knows, frankly? The wise man knows he knows nothing when it comes to influenza, so you always have to be cautious in speculating.
Ed Sussman with MedPage Today. So how do we approach, what kind of recommendations would you make the next time we have a novel pandemic as far as inoculating people with, or vaccinating people with a seasonal flu vaccine?
Well, I think that what we know about influenza is there's many commonalities across influenza strains. Influenza typically behaves like influenza, but there is a lot that we don't know as well, and we need to stay humble in that way. And I sincerely hope that at the first glimpse of a novel virus establishing sustained human-to-human transmission, the first question we will also be asking is, what about seasonal vaccine effect on that risk? And that studies will be implemented rapidly and early on. That, of course, means having rapid response research infrastructure in place to do those kinds of studies. But the recommendation would be to assess that as rapidly as possible. And the ferret model is an excellent model for, it's not perfect, but it is an excellent model for assessing that. And I would hope that more of these ferret experiments or animal experiments would be done at that time, but also, frankly, in advance. My co-investigators and I really sincerely hope that others will do this study, because this isn't the end of the story. This isn't concluding it. And it would actually be, I think, preferable to have groups outside of ourselves doing it.
May I interrupt you with a sort of follow-on to that issue, which is, I do understand, I think, why you would have looked at this, I don't know, this mode of interaction. You looked at the trivalent vaccine from 2008 and 9, because that was the phenomenon you saw in the human population, and you measured that against an H1N1 challenge. But isn't it possible for you to more systematically look at different vaccines, different seasonal vaccines, and different influenza challenges, so that you can maybe get a sense of how robust this finding is? That is it only H1N1 that would trigger a worse outcome, or are there other pandemic or different enough flu viruses that would trigger the same response, or maybe in some cases you would get a more protective response, and then you would begin to bracket the overall effect of these observations?
Yes,
Does that make sense?
It totally makes sense, and I completely agree with you. This is the starting point, I would say. There are many things that we would have learned from this study that we would like repeated and enhanced. So for instance, it was clear that by day 14 the ferrets were recovering, so we should be focusing then on what's happening at day 5 after challenge. I would increase the number of ferrets at day 5, and I would look at a number of different vaccines and different subtypes. So as I say, this is a clarifying first step. And what I'm hoping is that other scientists, other investigators will follow up with additional studies, additional enhancements on this. And I encourage that and ask for it, actually, that other investigators, other scientists will follow up and do studies to further clarify. There's so much still we have to learn about the immunologic interactions between seasonal viruses and pandemic influenza. And as I say, I think when we pick up a signal like this, with our immunization program, public health has a moral and a scientific duty to follow up to try to understand it better. This is a good first start, but there's more study that needs to be done.
Okay, good. We have one more question from someone in-house. Yeah.
Hi, Karthik Vishwanathan from Vistara. My question is that you said that there are two possible hypotheses for the observation that you saw in both humans and ferrets, the direct and the indirect effect. I'm trying to understand how you would put the indirect effect or the infection block hypothesis in the context of your results in ferrets, where I presume they would all be naive, right? So
I had a hard time hearing that.
We're both having a hard time hearing you. Sorry.
I guess my question is, how would you reconcile the indirect effect hypothesis in the context of naive ferrets, which I presume both the control and the experimental group would not have been exposed to any infection?
Okay, I think I understand. You're asking how do we generalize our findings from naive ferrets? By that you mean those who had not previously seen influenza infection to the human context where most humans have seen influenza before and are not naive.
Especially in the context of the infection block hypothesis.
Right. Okay. Well, what we did was we gave the ferrets 2 doses of vaccine, and then 3 weeks later we challenged them with pandemic virus. We had wanted naive animals to be included in the study. Most people work with naive animals. However, when we did the antibody testing on the animals, what we discovered was, and I think this is a common problem now with ferret experiments, they were not naive. They had, in fact, been infected previously, all of them, with influenza viruses. And in fact, it was an H3N2 virus. It was heterologous infection that they'd had previously. That's the benefit of having done a randomized controlled trial. The titers, the antibody levels in the vaccine and the placebo group were comparable. So the ferrets were not different, but they had been previously exposed to virus. They had been previously primed. And that's in fact necessary in order to get a good antibody response to vaccine in ferrets. So they were not naive. That has 2 effects. First of all, it better replicates the human experience. Secondly, it would tend to dampen any difference that we would otherwise find between the vaccine and the placebo group, because the idea being that if they have previously been infected, they may actually have some cross-protection, cell-mediated immunity from that previous infection. But the reason that I say it favors a direct vaccine effect is, notwithstanding that prior history, following 2 doses of vaccine and then pandemic H1N1 challenge, the vaccine group fared less well.
Okay, good. I think we need to wrap things up there because of our time constraints. And I see no further questions in the room. I think we've learned a lot about the response of ferrets to the pandemic H1N1 flu challenge, and we've also learned that there are no naive ferrets in Canada.
Well, that's not true.
That's really an important finding, I think. But seriously, I want to thank Danuta Skowronski of the BC Centre for Disease Control in Vancouver. We have another press conference coming up in about 25 minutes. I'm Jeffrey Fox. Thanks for tuning in to the first ICAAC Live press conference. There will be more today and on the next 2 days. Thank you.
Explore the fundamental role of microbes in the natural history of our planet with Microbes in Evolution, the world that Darwin never saw. Available at estore.asm.org.